Hi, everybody. I want to do a PowerPoint with you for week 8 for online microbiology this fall. And so I've prepared it using Zoom so that I can talk as you look at the PowerPoint slides. And so bear with me as this is the first time I've recorded a Zoom meeting and had a PowerPoint along with it. So hopefully it will be better than my other video where my cat jumped up on my papers and interrupted the flow of the lecture. So I will share my screen and then we'll go through the adaptive immune system, which is super cool and super interesting. And I've taken all the really important concepts from the chapter and highlighted them here in this PowerPoint so that when you do go back and read through the chapter, you'll have a better understanding of what it's about because the chapter jumps around a little bit and covers different topics. So it doesn't read very nicely and it doesn't flow very well in this particular textbook. So hopefully this will help. All right, so I'm going to share my screen. All right, hopefully everybody can see this. I'm going to hide my face because that's distracting. Okay, so we're covering the adaptive immune system. And this is chapter 15. And there are two important kind of features that characterize adaptive immunity, or some people call it specific immunity. That is specificity, meaning that it responds to specific protein signatures on the surface of different antigens. So unlike the innate immune system, which sort of recognizes these PAMPs or the pathogen-associated molecular complexes, these are very, very specific to each individual pathogen that might be encountered. And then there's also the component of memory. The lymphocytes that are involved in the adaptive immune system are able to remember or recall the first encounter with an antigen. And then if you get exposed to that pathogen or antigen again, you're going to have a really rapid response. And then it will take care of that before you even realize you might have been exposed again. So that's kind of the beauty of the adaptive immune system, is this memory function. There are a couple of important cell types for the adaptive immune system. The first group are the lymphocytes. We have T cells, which, remember, all the lymphocytes are produced in the bone marrow, but the T cells specifically mature in the thymus. And these cells are T helper cells, T killer cells, which are also called cytotoxic T cells, meaning cell and toxic. And then T regulatory or suppressor cells are the T lymphocytes. Then we have the B lymphocytes. These are made and mature in the bone marrow. The B cells will become activated and turn into plasma cells. These cells are the ones that make the antibodies. Generally, people say, oh, B cells make antibodies. Well, yes, they do, but only when they're activated and become plasma cells. And when you have cells making antibodies, which are these secreted proteins that can do all kinds of cool things, we call that humoral immunity, because the antibodies are secreted into the bloodstream. And for historic reasons, the bloodstream is always sort of referred to as the humor, the humoral part of the body, because of the humors and things like that people would talk about before they had a better understanding of how the body worked. The other group of cells that are important here are the antigen presenting cells. And there are 3 of those in our body. We have macrophages, dendritic cells, and the B cells themselves can present they can present antigens. I'll probably talk most about dendritic cells, but I do have an example of B cells, but it's all pretty much the same process of how the antigen is presented. And you learned in the last chapter about phagocytosis. And so in order for an antigen presenting cell to present the antigen to other cells of the immune system, it undergoes phagocytosis to take up the pathogen. Then it processes the pathogen by fusing the antigen in the vacuole with the lysosome. And then those proteins are broken apart. And then they're further processed in the Golgi apparatus and then presented on the surface of the cell. So that's how the antigens get presented. So an antigen is any part of a pathogen, and it can also be a foreign substance or pollen or laundry detergent molecule or cat dander or something like that, that will induce an immune response. And the antigens themselves are specific molecules on a pathogen. They're not generally the entire pathogen themselves. And this is why sometimes we can be infected with similar strains of the same thing, like similar strains of the flu or similar strains of a cold virus. It's because the immune system is so specific that it's only going to respond to part of the pathogen rather than the whole darn thing. The epitope, this is a term that you should probably know. The epitope is the part of the antigen that is specifically binding to the immune cell receptors. And remember, anytime I talk about a cell receptor, I'm talking about a protein that's sort of sticking off of the surface of the cell that's able to interact with the environment. There's another important feature of the adaptive immune system, and that is its ability to recognize self versus non-self. So this system that allows the immune system to do that is dictated by the major histocompatibility complex. And we have two MHC molecules that are involved in this process. So every cell in our body that has a nucleus carries the MHC class 1 protein on its surface, like this. And every cell that can present antigens, that is the macrophages, dendritic cells, and the B cells, carry class 2 major histocompatibility complex proteins on their surface. All right, so if you're an antigen presenting cell, you will carry both of these. If you're a mature red blood cell, you won't have any of these. If you're a liver cell, you'll have a major MHC1 molecule. If you're a toenail producing cell, you're going to have an MHC class 1 on your surface. So B cells are, we'll talk about them first. They need to respond to an antigen, and they can do that either through the help of a helper T cell or directly by interacting with the pathogen itself. So here's something that the book goes into a lot of detail about, and I feel like it's really complex and it's easier to understand after you understand everything else that the book talks about. But the thing that you need to keep in mind about how B cells work is that every single B cell, when it's matured, will have a different sort of receptor pattern on the outside of its cell surface. And so it's estimated that we have about 10 billion unique B cells that are circulating in our body at any given moment. And this is because of this process of genetic rearrangement that happens when the B cells are maturing, and those proteins that are expressed on the surface are all different. And it's sort of this way that our body makes up all these different versions of B cells so that we can bind to different things that might be pathogenic to us or whatever. So these B cell receptors are all different in our population of naive B cells. And what I mean by naive is, these are the B cells that have not yet been presented with an antigen or a microbe or something like that. They're just floating around in the body waiting to bind with something and become activated. So in this cartoon, I've drawn a microbe. Here we have streptococci, like strep throat. And this receptor on the surface, we'll call it an immunoglobulin receptor, abbreviated IG. And remember, MHC class 2 is on the B cell, and I've left out the MHC class 1 because it's not involved in this. So the microbe is bound by the IG receptor on the B cell, and then it's processed inside the cell through the process of phagocytosis. And then the protein part of the antigen that's been processed is secreted and expressed on the surface along with the MHC2 molecule here. Then along comes a T helper cell, or a helper T cell. I interchangeably use that. These cells have a T cell receptor, abbreviated TCR, and helper T cells also have this other cell protein called CD4, and that makes them, they'll either become a helper T cell with a CD4 protein, or they can become a regulatory T cell, which I'll tell you about in a little bit. Anyway, so here we have the B cell. It's presenting the antigen in this case, but the helper T cell is important for the B cell to become activated. And most B cells require a helper T cell to be active. So these two cells get close enough together so that the B cell can actually sort of present the antigen to the helper T cell. That helper T cell says, oh boy, you've seen some rough stuff today. And so then it starts to secrete growth factors for the B cell and some other cytokine molecules. So there's this whole class of molecules that get secreted by the immune system cells called cytokines. And one group of these are called interleukins. And interleukins are the chemicals that are produced. They're like chemical messengers that are produced by the leukocytes, by the white blood cells. And so they're interleukins. And so the helper T cell will produce these molecules, and that will cause the B cell to become activated. Once that B cell is activated, it will undergo mitosis and make a whole bunch of copies of itself, because now it has this Ig molecule on the surface that's important because the body's now under attack by a foreign invader, and it needs to make more of these. And so that mitosis and that process of mitosis is called clonal selection. And so this B cell, some population of these B cells that have undergone mitosis will become memory B cells, which is awesome because that means they'll circulate in the body for years, just in case the pathogen comes back. And then another fraction of these cells become the plasma cells, which are the cells that are actually secreting the antibodies. And here are the secreted antibodies. This is a photo from your book showing the same thing I just showed you. So, but in this case, we have a different antigen presenting cell. We have the dendritic cell, which is usually the cell most textbooks show as the antigen presenting cell these days. Back when I was in college, we didn't really know about dendritic cells as well. So it was always just some antigen presenting cell, like a macrophage. But anyway, what the classical example is that you have a dendritic cell. It shows the helper T cell, the antigen, then the helper T cell goes to the B cell, and the B cell will then become activated because of all those cytokines and interleukins that are produced. And then again, it will become, some of them will become memory B cells, and the others will become plasma cells, and they'll be secreting the antibodies into the bloodstream. This again is called the humoral immune system because the proteins are going into the blood system so they can travel throughout the body. And then those antibodies will do the various things that antibodies can do. So antibodies are also called immunoglobulins. That's just a fancy term for a protein that does something with the immune system. And they have two functionally distinct segments. And the part that's really important are the outermost parts called the antigen binding sites in the variable region. These are the parts that differ for every single B cell that's produced. Then there's the crystallizable fragment. These can play a role in complement binding, which is pretty much the only important thing they do. They don't vary as much as the variable regions, obviously, because these arms up here need to be different to be able to recognize different antigens that come into the body. So once these antibodies are produced, they can do a number of different things. So probably the most common thing they do is neutralization, meaning that they go and they bind to the antigen, or they bind to an incoming virus or something like that, and stops that pathogen from being able to bind to and infect cells in the body. Antibodies can also do something called opsonization, in which they bind to the pathogens, and then the pathogens are easier for the phagocytes to see. So the macrophages can more easily bind to and phagocytose those pathogens if they've been opsonized by the antibodies. Agglutination is another thing that antibodies do, in which they will bind to a whole bunch of different, say, bacterial cells and kind of make them clump up, which again makes it easier for the other cells of the immune system to come in and do phagocytosis. Antibodies will also bind to complement. That's what complement fixation means. It just means it's binding to complement. And you'll remember from chapter 14 that complement, the major role of complement is that it will all these little proteins will bind to a bacterial cell or a virus infected cell and poke holes in that cell to kill it. So it's just another way to kill the pathogen that's in the body. Another thing that antibodies can do is precipitation, in which they bind to antigens and kind of keep them out of circulation, out of the blood system. And here's just what I said in another form. So we have the bacterial cells are tagged with antibodies in terms of making them easier to see for the immune system. And here's opsonization. So we have the macrophage, and now they're better able to bind to these bacterial cells and then get engulfed. And then here's neutralization. So here we show viruses that are bound with antibodies. If a virus is bound on its surface with all of these antibody proteins, the receptors on the viruses that need to attach to the host cells are no longer there. So then the virus can't bind and infect the body cells. Then we have agglutination, where we've got cross-linked bacterial cells. And here we've got complement fixation. And I like this little picture here. It's showing the complement proteins causing these cells to burst open like little fountains, like geysers coming out. And then precipitation here, we've got the antigen molecules just get taken out of circulation by the antibodies. So antibodies can play a lot of different roles in the body, but the main purpose of all of them is to get rid of the pathogen, the bacteria or the virus or whatever it is inside the body. There are 5 classes of antibodies, and they're produced in different orders as well, which is kind of interesting. And I think it's hard to teach this because we don't really understand the ways in which this all works in a big holistic way. We kind of understand how these work in specific instances. So IgM, I'll start with that. This is this big pentamer. There's 5 of these immunoglobulin proteins kind of linked together. This is the first antibody that's produced by any activated plasma cell. And it's big, and it cannot cross through the placenta. And when we talk about blood types, like if you have A type blood or B type blood or AB blood or O blood, we're talking about antibodies in the bloodstream that can recognize the opposite types of blood. And so those antibodies that recognize the different blood types are IgM. And that's a good thing, because if they were the smaller antibody molecules, that would be a bad situation for a baby that was developing in a mother's womb who had a different kind of blood type. And so the IgM molecules can't cross the placenta, and so they can't bind to and attack the developing fetus. The Rh factor is a different matter, and we can talk about that another time. IgG is usually produced second, and it's the most common antibody class you find in the blood. It's probably because it's the longest lasting, and it's also called gamma globulin. You might hear that just because of the G. And this one can cross the placenta. So you can look this up and understand how it works, but when an Rh negative mom, so she's got, say, O negative blood, has a baby with, say, A positive blood, that's not a big deal if it's the first baby. But the second baby, that might be a Rh positive baby, would trigger response by the mother's antibodies that were produced if she was exposed to the blood type of the first baby during the birth process. And so she would have antibodies produced, and that could attack the baby and called hemolytic disease of the newborn. Well, these days they know about that. So usually what they do is, if you're an Rh negative mother, they give you RhoGAM, which is a blocking agent, which will block your immune system from being able to respond to an Rh positive antigen. So that's what that's about. All right, so IgG is most common, longest lasting, found in the blood. It's the one we do a lot of testing for if we're looking for antibodies against something. Then there's IgA. This one's is associated with body secretions like tears and saliva, breast milk, etc. Then we have IgE. This one is involved in allergy responses as well as parasitic infections. And then we have IgD. This is found in the B cell membrane, and it may act as a B cell receptor, BCR, B cell receptor, but we don't know the exact function. So It's there, it must be important, but we're not sure why. When we produce antibodies in response to an infection, there are two responses, the primary response and the secondary response. The secondary response is also called the anamnestic response, which is a term you need to know. So the first time you're exposed to the antigen, there's a latent period of about 2 weeks. Then you start to produce a whole bunch of IgM. And then after that, you produce even more IgG. And that's longer lasting, say within the first month. And then over time, you've cleared the infection, you're no longer sick, but then you get exposed to that antigen again. Well, you make the IgM pretty much in the same way, only notice it's happening much sooner, a day, starting to ramp up maybe at day 2 or so. And IgG production is really high at this point, and it lasts much, much longer because of the activity of those memory B cells producing so much more of these antibodies than during the primary response. So it's pretty fantastic. And it's because of this that vaccines work so well, because we can give somebody a vaccine and induce a small immune response here, and then we can give them a booster and get a much, much bigger response. And so that's why a lot of vaccines, including the COVID vaccine, is something that requires a booster, because we want to make sure you have a whole bunch of these antibodies circulating in your bloodstream to bind to any virus that you might get exposed to. There's another thing you might have heard about called monoclonal antibodies, and these are kind of an interesting technological advance that came about in the 1980s. And so what they are antibodies that you can produce in a laboratory. And these are used for treating diseases, sometimes diagnosing diseases. They use these when they make like any sort of diagnostic test, like so for a rapid strep test or a pregnancy test or something like that, they make these antibodies in a laboratory and then they put them into filter paper. So then you can, test them against urine or blood or whatever, or throat culture swabs. So how these are done is you take a mouse and you inject it with an antigen. And just like our bodies, the mouse bodies make antibodies. And then we have these myeloma cell lines that are grown in culture. So a myeloma is a kind of cancerous cell line. And a lot of cancer cell lines do this really interesting thing in cell culture where they just grow and grow and grow and grow, and they become immortal, so to speak. And so they have these immortalized cell lines that they mix with the antibodies and they fuse them together. And how they do this, they can do this with adding different chemicals. They can use an electrical current. They just kind of get these plasma cells and the cancer cells to fuse. And then they produce these long-lived sort of antibody producing cancer cells, which sounds so science fiction, but it's so science nonfiction. It really works. And then they put them in these little wells, and then they test them for reactivity against the antigen that they were starting with. And then they collect the antibodies that are produced by this hybridoma. It's because it's a hybrid between two different cell types. And then they can isolate those antibodies and use them for various things. So I think it's just such an interesting thing that somebody somewhere thought, well, I wonder if I just mix these plasma cells from a mouse with these cancer cells and see what happens. And lo and behold, it worked. And now we can get antibodies made for treating all kinds of things. So like say you early on in the COVID pandemic, they were using monoclonal antibodies against COVID to treat people who are severely ill from the disease and things like that. And so this can be really beneficial to folks if we don't have another ready treatment for a particular disease. They'll turn to monoclonal antibodies for that. All right, so we're going to switch gears and we're going to start talking about T cells. And T cells are the other arm of the specific immune system, and they're generally associated with cytotoxic or cell-specific immunity. And so if they are activated, they can directly kill virus-infected cells as well as cells that are damaged or cancerous. And there are 4 groups of T cells. There are the helper T cells, and they're going to have a CD4 T cell receptor on their surface. Killer T cells will have a CD8 T cell receptor on their surface. All that means is that there's this protein on the outside of the cell, and it's got a different shape. So 4 is going to have a particular shape, 8 is going to have a particular shape on the outside of that cell. Then we have the suppressor cells, and the suppressor T cells are sometimes called regulatory T cells or Tregs. These actually have CD4 receptors on their surface, and their job is to keep the immune system from going out of control. So they will sort of make sure the immune system stops when everything is taken care of, when there's no more virus in the body, or there's, the infection has gone down or whatever. And then we have the natural killer cells, which we talked about in chapter 14. And these are involved in the innate immunity, which they're phagocytes essentially. All right. So T cells will get activated a lot like the B cells do, but the antigen is presented with an MHC complex, just like the T helper cells. You already know that because we just talked about it. So the CD4 T cells are activated by antigens that are presented on the MHC class II receptors, right? So you have to have an antigen presenting cell here. Whereas CD8 cells, which become killer T cells or cytotoxic T cells, these guys are activated by antigens that are on the class I receptors of the antigen presenting cell. So that's pretty interesting because that means that these class I receptors, if there's an antigen on those surfaces, that means that that cell, that antigen presenting cell knows that it's like a virus, and it can present it on a different receptor. So then it tells a CD8 cell to become activated instead of a CD4 cell. So it's a really interesting difference that goes on. And so here's a picture. I don't know if this is from our textbook right now or the face-to-face lecture textbook, but here we have the immature lymphocyte. It will mature in the thymus, and it will become either a CD4 cell or a CD8 cell. Notice the different shapes of the receptors. Then we have an antigen presenting cell, and for the CD4 cell, it's activated by MHC2 presentation, and it undergoes mitosis, and it produces memory cells just like the B cells do. And then we have the activated portion of the cells, and these cells, we call them effector cells, and they're going to go around into the body, circulate in the lymph and the bloodstream, and look for virus infected cells I shouldn't say virus infected cells. They're going to go and they're going to circulate around the body to find B cells to activate, as well as other things that need to happen. On the other side, on the right side, we have the CD8 cell. It's going to be activated with presentation of the class I molecules, the class I MHC here. And these are going to produce some memory cells. And then these effector cells are going to circulate in the body and look for virus infected cells. And again, the suppressor T cells, those are going to be triggered over here, and they're produced at the same time helper T cells. And their job is just to kind of do surveillance and make sure the infection gets taken care of. And then they turn off the immune response when necessary, so that we don't have this sort of hyperactive immune response to nothing. All right, I'm going to switch gears again and talk about different types of immunity. So here we've got 4 classes of immunity. We have active immunity, and this is when you make your own antibodies and make your own memory cells. This takes time, and it can be very long lasting. Then we have passive immunity. This is when you have preformed antibodies are given to you. They're donated to you, usually through an IV. They don't create memory, but they will act immediately, and it's short term. So passive immunity, when you have preformed antibodies, this would be antibodies that are given to you through breast milk when you're a child, when you're an infant, as well as, if you had a monoclonal antibody injection, if you were ill with something and you got that. So that would be passive immunity. It means you're not making the antibodies yourself. It means you're getting them from somebody else. You also have natural immunity and artificial immunity. So natural immunity, you acquire that as part of your normal life experiences with the different infections and things that you get exposed to. And an artificial immunity, you get that through vaccines. So then you can have different groups of these, 4 types of immunity. You can have natural active immunity, which is getting an actual infection. That one's easy. Then natural passive immunity, that's when you get antibodies from the maternal bloodstream as you're developing in utero, or it's passed through the breast milk. Then you have artificial active immunization, that's vaccination. The person who made these slides changed it, I think, to make it more clear. So this is when you get, part of a microbe injected into your body and you make your own antibodies, but it's artificial because it's not a natural infection. And then you have artificial passive immunization, and this is where you get immunotherapy. So you're getting a monoclonal antibody, or maybe you're getting like antivenin or something like that because you had a snake bite. A lot of people are very interested in how vaccines are made these days because there's a lot of talk about vaccination. And so I'm going to tell you about a few of these types of vaccines. So we have killed whole cells or killed or weakened viruses. So these are generally what's in the vaccine. You can have antigenic molecules that are derived from the bacterial cells or viruses. So it could be like parts of a bacterial cell or parts of a virus that are used. And then we can also have genetically engineered microbes or parts of microbes. So they can be natural or genetically engineered. And then also the very most recent type of vaccine that some of us have had some experience with are the mRNA molecules and the mRNA vaccines. And so we'll talk about that too. Attenuated viruses or attenuated vaccines versus a killed vaccine, they're very interesting. So one prime example of this is the flu mist versus the flu shot. So in some years you can get the flu mist, and the flu mist contains attenuated flu viruses. And so they're alive, if a virus can be alive. So I should say they're active. They can cause an infection, but they won't really cause the disease. So they cause sort of a little bit of an infection in your upper respiratory tract, and then it goes away. It's a really weak kind of thing. For a lot of people, it just feels like they've got allergies or a really mild cold. Versus the killed vaccine, which is a completely inactivated virus. So when you get the flu shot, that's just pretty much a boiled virus. It's completely inactive. It can't cause any kind of infection, but it has all of the important proteins and things that can generate an immune response to that virus. So whole cell vaccines, so you have the killed or inactivated virus, or the live and attenuated virus. You have the antigens. And this is just sort of a picture of exactly what I just said. So then we give the vaccine, and people will generate their active immunity against this artificial vaccine. Then we have vaccine from microbe parts. So this can include capsule proteins like pneumococcus, that's Streptococcus pneumoniae, or meningococcus, that's Neisseria meningitidis bacterium. There's also surface proteins, like so anthrax. Most of us don't get an anthrax vaccine unless we're in the military. But if you go into the military, then you often get anthrax. But a lot of us have had a hepatitis B vaccine if we work in healthcare. Then there's also exotoxins like diphtheria and tetanus. And so every, 10 to 15 years, we should get a tetanus shot. And that's just this exotoxin from the tetanus bacterium that is given to you. So should you step on a rusty nail or whatever, you don't develop tetanus. Then there's recombinant vaccines. So remember, anything that is recombinant means it's genetically engineered. And so here we have a virus, and they've put a part of that virus into a plasmid, and then they put that into a yeast cell, and then the yeast cell makes that protein that is then purified and used for the vaccine. So this is a hepatitis B example of how they do that. So it's just a way, this can make a whole bunch of protein for that vaccine in a really cheap way. And it's also safer because you're not growing up a whole bunch of virus just to purify virus parts, because that can be really expensive and also dangerous for the people who have to do that. And then the mRNA vaccines. So to understand the mRNA vaccines, you should remember the central dogma, right? And that just tells us that if you have DNA, it gets made into mRNA. And if a cell recognizes mRNA, then that mRNA gets translated into protein. So how this works is people took the protein, the gene to make the spike protein, and they generated mRNA from that gene, right? So they probably cloned the spike protein, expressed it in a cell, whether it was a yeast cell or a bacterial cell or whatever, and then isolated the RNA and then encapsulated it into this lipid nanoparticle. And that's really the key for the mRNA vaccines. The mRNA doesn't matter that much, but the lipid nanoparticle is the part that is so crucial in the ability of these vaccines to work. So they get these mRNA molecules in a lipid nanoparticle. They put them together in the vaccine, and you get it shot into your arm, which is where you usually get most of your vaccines. What happens is it goes into the dendritic cells, which are circulatory antigen presenting cells. So they'll be picked up. They'll be in your arm. Probably they'll come around as well because you have a small wound from the injection, and that triggers a small immune response because you've got this inflammation response from the wound of the needle. Anyway, so the dendritic cells come, and they will bind to and undergo endocytosis to take in the lipid nanoparticle. They'll process it, and in the process of processing it, they'll translate that RNA that was in the vaccine, and it will make it as a protein. It will turn it into protein and present it on the MHC class II molecules here. And then that will activate B cells to produce it will activate the T helper cell, it will activate the B cell, and it can also, if it's expressed on an MHC1 class molecule, it can express the T killer cells. And again, just like we've talked about, the B cell and T cells work together to make memory cells and plasma cells. I guess activated B cell is the same thing as a plasma cell. So then they go on and they produce antibodies, which will exist in the body for some period of time to neutralize any virus that might get into the body. And then we have activated T cells will be produced as well in this situation, and they can go throughout the body, and you'll have a group of memory T cells here that will remember if that virus happens to get into the body. And so that's how the vaccines work, is that we have all of these cells that are activated and ready and waiting should we encounter that particular antigen in the body. So the last slide of this talk is talking about herd immunity. And you can't talk about vaccination or epidemiology without talking about herd immunity. And so a lot of people have talked about herd immunity this past couple of years because of the COVID pandemic. And so this is showing each of these hexagons, I guess, as a person, and this is the pathogen. And so if you have no immunity in the population, you're going to have a whole bunch of people who get sick right away. Then we have people who can recover, and those are the hashed lines here, and they can no longer transmit the disease if they've recovered and they can't get infected again. And this can happen for a period of time. Sometimes it's shorter, sometimes it's longer. It really just depends on each person's immune system and the nature of the pathogen. And here we also can have vaccination, which will increase herd immunity to a higher percentage, and it can stop the pandemic sooner. And it generally reduces the death rate as well. So once we reach herd immunity, which means that you have about 55 to 82% of the population who can no longer become infected by the pathogen, that's how we define what herd immunity is. And it's different for every pathogen, and we never really know until after the fact. Anyway, so this means that you just have a whole bunch of people who are vaccinated, and then you have a bunch of people who aren't. But what happens is, even if these people are infected, they're not going to pass it to that many people because it can't pass anymore because these people have active immune responses against the pathogen. And then the pathogen can no longer spread throughout the whole population. So herd immunity is something that deals with how populations respond to infectious disease rather than just individuals. And once a certain number of people get vaccinated, usually the diseases are no longer considered critical, like measles, for example. We have episodic outbreaks of measles all the time, or even Bordetella pertussis. And sometimes people don't get vaccinated because of whatever reason, and a lot of people do get vaccinated, and so we get this herd immunity, so we're not inundated with measles cases or Bordetella pertussis cases. And hopefully in the future enough people will get vaccinated against COVID to stop the spread of COVID to the point where we have to worry about having enough hospital beds for ICU patients and that sort of thing. So that's the end of my PowerPoint presentation. Please email me with questions if you have any questions regarding chapter 15 or anything else in the course. And with that, I will just end there.